Dear students and my teacher friends, Namaskar. I welcome you to the presentation lecture on digestive enzymes. Now, before I start this presentation lecture, I would like to tell you a few things regarding this lecture. One thing that I would like to tell you that you can skip the preparation of this topic. Once again, I am telling you, you can skip the preparation for this topic, digestive enzymes. But there is one condition. There is a one condition in that you can skip the preparation of this topic, digestive enzymes. Now, what is the condition? The condition is that you should have prepared the topic of digestion in the mouth. Then you should have prepared the topic of digestion in stomach. You should have prepared the topic of digestion in small intestine. Then you should have prepared the topic of digestion of carbohydrate, protein, dietary fats. You should have prepared the topics of absorption of carbohydrate, dietary fats, and protein. In that case, you can skip the preparation of this digestive enzyme. Now, as far as I am concerned with this topic, so you know that I have already delivered all the lectures: digestion in mouth, stomach, small intestine, then the digestion of carbohydrate, protein, lipid, absorption of carbohydrate, protein, lipid. All these lectures I have already delivered, and the lectures are available on my channel. And here, for this is actually the summary of digestive enzyme, this table. So this table is very important for you because it is summarizing all the digestive enzyme. The table, this is the entire table. Once again I am showing you. And one more table is there. And I'm not discussing these things because you know that I have delivered these separate lectures for all this topic. And this is the summary. Summary of carbohydrate digestion, absorption, protein digestion and absorption. So this information is for you to prepare. Now both the table, if you want to prepare by your own, then I would say tata bye bye. Lecture is over. Now if you want to listen, I am explaining the table to you. Now what this table is. So here, as you can see in the table, there are 4 columns. In the first column, the name of the enzyme, digestive enzyme name is written. Then in second column, source, from where the enzyme is secreted, from where the enzyme produced. Then the substrate on which the enzyme is acting, and the product of the digestion in the last column. So one by one we are discussing this. First, enzymes of saliva. Now saliva is secreted by salivary glands in the mouth. So in the saliva, salivary amylase enzyme is there. Salivary amylase digesting the starch, and the product of starch digestion, maltose, maltotriose, alpha dextrine. Then in the mouth there is a tongue, and in the tongue there is a lingual gland. Lingual gland secrete or produce so saliva once again, and the saliva is having the enzyme lingual lipase. Now this lingual lipase digest little bit, very little fat and oil digestion takes place with the help of lingual lipase. So triglyceride, they are converted into fatty acid and diglyceride. Then gastric juice, stomach. Now we know from the stomach chief cell, the pepsinogen, which is in inactive form, is activated by HCl. Then it is converted into active pepsin, and this active pepsin converts the remaining pepsinogen into active form, that is the pepsin. Now what this pepsin is doing? So pepsin digests protein, and proteins are converted into peptides. Then cheap cell, gastric juice, gastric lipase, the name of the enzyme. Gastric lipase act on triglyceride, fatty acid and monoglyceride are produced. Now pancreas. In the pancreas, acinus, pancreatic. So we have studied the histology of pancreas, and in that we have learned about the pancreatic acinus. So in the acinus there are the acinus cell, and this acinus cell they secrete the pancreatic juice. Now the enzymes of the pancreatic juice, one is pancreatic amylase. Pancreatic amylase digest starch. So starch is converted into maltose, maltotriose, alpha dextrine. Then trypsin. So it is in an inactive form, trypsinogen, and trypsinogen is activated by enterokinase, and trypsinogen is converted into active trypsin. Now what this trypsin is doing? Trypsin digesting the protein and the formation of peptide. Chymotrypsinogen activated by trypsin. So active form chymotrypsin. Chymotrypsin act on protein, digest the protein. Proteins are converted into peptide. Then inactive proelastase activated by trypsin. So active form elastase. Elastase digest protein. Proteins are converted into peptide. Then procarboxypeptidase, inactive form activated by trypsin, active form of enzyme carboxypeptidase. Where this enzyme is acting? So it is removing the amino acid, terminal amino acid from the carboxyl end, COOH end. So amino acids are released and the peptides are formed. So digestion of protein, in that the carboxypeptidase is involved. Pancreatic lipase secreted by the pancreatic juice is secreted by pancreatic acinus cell, and in the pancreatic juice pancreatic lipase enzyme is there. This enzyme act on fat and oil, triglyceride. So after the digestion, triglyceride they are converted into fatty acid and monoglyceride. Then the nucleases, ribonuclease act on RNA. So RNA digestion yields ribonucleotide. Then deoxyribonuclease, DNA acts on DNA. So DNA deoxyribonucleotide. Digestion of DNA produces deoxyribonucleotide. Then here brush border enzyme, where they are present, microvilli, plasma membrane. So the mucosal epithelial cell of small intestine, that are the enterocyte cell, and this is the apical membrane of the enterocyte cell. One more I am drawing. So this is the apical membrane of enterocyte cell, and this apical membrane and this finger-like projection, they are known as the microvilli, and all the microvilli together, they look like the bristle of the brush. So that's why this border, this apical membrane, is known as the brush border. And on the surface of this brush border, enzymes are present, and these enzymes are present, and these enzymes are also involved in the process of digestion. So these are the brush border enzyme. One is alpha dextrinase. Alpha dextrinase act on alpha dextrin converted into glucose. Maltase act on maltose. Maltose is converted into glucose plus glucose. Sucrose, glucose plus fructose. Lactose, glucose plus galactose. Enterokinase. Now this enterokinase, this is trypsinogen inactive form. So due to this enterokinase enzyme, trypsinogen is converted into active form, that is trypsin. Then peptidase, aminopeptidase. So aminopeptidase is breaking the bond in the polypeptide at the amino end. Amino end means NH2N, where the amino group is present. So that bond, that peptide bond, this aminopeptidase is breaking. So the terminal amino acid, where the NH2 group is there, so that amino acids are released. Okay, so there is a formation of amino acid and the peptide. Then the dipeptidase act on digest the dipeptide. So peptide bond is broken down by this dipeptidase, and there is a formation of amino acid. Then nucleosidase and phosphatase, they act on nucleotide. So digestion of nucleotide, ribonucleotide and deoxyribonucleotide. So they produces nitrogen base, purine and pyrimidine, adenine, guanine, cytosine, thymine, uracil. You know the names of the nitrogen base. Then the pentose, either the deoxyribose sugar or the ribose sugar, and the phosphate. We know phosphate is the phosphoric acid, that is H3PO4. Okay, so all these are produced. So this is how. So basically, in this topic of digestive enzyme, what you need to know, the source, which is the organ is involved for the production of enzyme, where the enzyme is acting, which one is the substrate, and after the digestion, which are the product, that you need to know in this topic. And once again, I am showing you both the tables, this way and this way. So with this, we have completed this presentation lecture on digestive enzymes. I hope this presentation lecture will be helpful in your exam preparation and also in your studies. My name is Manish Koshti sir. I am from Ahmedabad, India. Bye bye. Namaste.